Okay, hello everyone. Good afternoon, good morning, good evening, depending on where you are in the world. But most of us I know are based in Africa. So hello. My name is and I am the current chair of the Education and Coordinated Training for H3Africa. And I would like to welcome all of you to this, what promises to be an amazing session for the H3Africa Fellow presentations. We have about 4 presentations today, and I'm going to kindly ask you that you engage, that you give your comments, and that you ask questions. And please do specify whatever question, or rather, who you directing your question to. So there's a Q&A where you can type in your questions, live Q&A section, or otherwise you can put it in the discussion forum, and I'll be checking both to see where the comments and questions are for each of our presentations. So what we'll do is all of these videos are all these presentations are pre-recorded. What I'll do is that I'll introduce each presentation as we go, but please do sort of type your questions in the Q&A and in the discussion forum as well. While obviously each presentation is going on, at the end, I'll invite all our presenters to sort of engage with you on each presentation. The first presentation is coming from Linda Forter, and Linda's going to be presenting on a novel variant in splicing genes causing SPG11 in a consanguineous malign family with hereditary spastic paraplegia HSP features. And without further ado, I'm going to hand over to Linda to give a presentation. Thank you.
Hello, everybody. My name is Linda Forter. I'm a student at the Faculty of Medicine and Dentistry of the University of Science, Techniques and Technology of Bamako. It is a real honor for me in occasion of 18th H3Africa Consortium to present our work concerning a novel variant in the splicing gene causing SPG11 in a consanguineous malign family with hereditary spastic paraplegia feature. Our presentation will respect this order. Hereditary spastic paraplegia or Swamper-Lorraine disease are heterogeneous group of neurodegenerative disorder characterized by progressive weakness, hyperreflexia, and spasticity at the lower extremity. Most HSP are inherited in an autosomal dominant or autosomal recessive pattern, but autosomal recessive cases are predominant in the Mediterranean and sub-Saharan Africa. Phenotypically, HSP are classified into pure, in which the main clinical feature are progressive spastic paraparesis associated with posterior colon or bladder involvement, and complex where additional neurological and non-neurological features are present. Hereditary spastic paraplegia type 11 linked to pathogenic variant in the splicing gene, which represent the most common form of complex autosomal recessive HSP, and exhibiting additional neurological symptoms such as cognitive decline, tinnitus, callosum, and cerebral ataxia. To date, only few genetically cases of SPG11 has been reported in sub-Saharan Africa, including one from Mali. Our objective is to clinically characterize malign families with hereditary spastic paraplegia and identify the underlying genetic defects. Our methods are patient and dual relative when through a thorough neurological examination after giving a written informed consent. Blood chemistry, brain, and spinal imaging were done to exclude common causes. DNA was expected for whole exome sequencing. Pathogenicity of putative variant was confirmed by in silico analysis. We investigated large consanguineous malign family, in which 3 cell being border show HSP future, represented by this figure. Black arrow show index passion, and star all those patients seen in clinic. Consanguineous marriage of two unaffected parents with showing recessive inherited pattern. Age at onset was 10 to 20-year-old with walking difficulty, spasticity, speech disturbance, and tremor that worsened gradually. Neurological examination found HSP symptoms such as spasticity and breath reflex in falling, extension plantar reflex, ankle trepidation, lower muscle weakness predominantly in the lower limb, and spastic gait were associated with cerebral ataxia signs such as rest and attitude tremor, dysmetria, allodotokinesia, dysarthria. Without of hypothesis diagnosis, recessive cerebral ataxia and recessive complex SPG. We have done HTLV1 serology because he causing tropical paraparesis mimicking SHP. It was negative. Cerebral tomography shows cerebral atrophy represented in lateral and dilating of lateral ventricle and cortical sulci. Whole exome sequence identify a compound heterozygote located on exon 30 and 36. The first variation is located on exon 36. It is substitution type mutation where cytosine was being replaced by theanine. Consequence of protein is replacement of amino acid arginine by codon stop, leading to a loss of function of the protein. This mutation has already been reported in the European population in 2 individuals. The second variation is located on the exon 30, where there has been an insertion of the theanine nucleotide. A protein consequence is a shift in the wedding frame of all the rest of the amino acid sequence, resulting in a loss of function of the protein. It is a frame shift mutation. This mutation has not yet been reported. We confirm this mutation by Sanger sequencing. The first variant is present in the mother and patient and not in the father. We conclude that this variant is from a mother. The second variant is present in the father and patient and not in the mother. We conclude that this variant is from a father. In conclusion, we report here a novel heterozygote frame-shift variant in spartaxin gene causing SPG11 in a consanguineous mammalian family, confirming the genetic heterogeneity in an African population. Special acknowledgement to all the study participants and the research team for the opportunity.
We're speaking about.
Hello, everybody. My name is Linda Fota.
I think
I'm
the video is supposed to be paused. There might be just speaking of another variant of. Okay, so thank you. That was technology failing us. That was a very calm sort of an educational presentation from Linda Foster. We're speaking about a novel variant in Spatian gene causing SPG in Malayan family. Without wasting any more time, I'm going to sort of move on to the next presentation, which is going to be by Goita Mudiboke, who's also available to answer your questions later. So please do type any questions or comments that you have for a specific presenter in the live Q&A. I'll appreciate that a lot. The next presentation is going to be by Goita Mudibo, as I said, and it's going to be on complex genomic rearrangements and individual telomere length in chronic myeloid leukemia. Go ahead and give your presentation, Goita. Thank you.
Hi, everyone. I'm Modiboke Goita from Mali, a pharmacy student. I'm part of Gida Landure's team. Today, I'm presenting a complex genomic rearrangement and individual telomere length in a chronic myeloid leukemia patient with a trisomy X. I'll follow the present plan, starting by an introduction, objective, methodology, result, discussion, and close by a conclusion. As an introduction, chronic myeloid leukemia is a myeloproliferative disease characterized by the presence of translocation 922 found in almost all cases. Around 10% of these patients show an atypical translocation, which can involve one or more chromosomes in addition of chromosome 9 and 22. Telomere shortening is correlated to CML stage and the outcome of patient prognostic. Therefore, less attention has been paid to the role of individual telomere length in the prognosis of CML. And it's essential to know the individual telomere length correlation with atypical cases involving the translocation 922. For that, the aim of the present study is to measure the telomere length on each chromosome arm of CML patient presenting atypical translocation. We screened 130 patients presenting CML. Among them, we found 28 presenting atypical signal fusion. We chose the atypical cases and we did the following protocol. After choosing the best metaphases spreading slide after cell culture, we did first multicolor fluorescence in situ hybridization MFIT, which stain all homologous chromosomes with different color and help us to characterize the chromosome abnormality. Then we did on the same slide on the same metaphases quantitative fluorescence in situ hybridization QFUS used to quantify telomere length signal. Penaprod stain telomere on each chromosome and with a red color, and pancentromeric prod stain all centromere in green used as internal control, and then we quantify telomere length signal. Atypical cases represent 25% of all the 130 CML patients that has been enrolled in this study. Among them, we found a unique genomic rearrangement that has been never described to our knowledge, a case representing trisomy X with a complex translocation involving chromosome 9, 22, and 16. This is a M-fish karyogram, and we can see at its right side is translocation involving the chromosome 22 at the top, color in red, a part of chromosome 9 in green at the middle, and a part of chromosome 16 in yellow at the bottom, forming a single chromosome and the trisomy X. This is a new discovery to our knowledge, and the patient presenting this translocation, we follow up the patient and we found the patient was resistant to the current treatment of tyrosine kinase inhibitor. And this is a QFISH picture. We didn't see well in DLP staining which chromosomes were involved in this complex translocation. In reason, we use M-FISH technique to characterize translocations and did a first classification and use that as a guideline to classify the same QFISH metaphases and then measure the telomere length. This is the graph of the average of the measurement of 20 metaphases, and we found that the short arm of chromosome X presented the longest telomere, while the long arm of the chromosome 21 presented the lowest telomere. After 10 months of imatinib treatment, we follow up the patient and we found that all the 3 cell lines were down, and we found atypical signal in 35% of leukemia cells and 65% cells were normal. That show the patient did a primary therapeutic resistance. Although a great number of complex translocations have been described in CML, to our knowledge, this genomic rearrangement with a trisomy 8 is the first to be described in CML. Therefore, this genomic rearrangement shows a primary resistance to imatinib, and the length profile of individual telomere of atypical and typical translocation are different. In conclusion, we found a unique genomic rearrangement in CML, a case presenting a trisomy X with a complex translocation involving the chromosome 9, 22, and 16. And we show that this novel variant is responsible for the primary resistance of tyrosine kinase inhibitor imatinib in CML. And we found that the length of individual telomere is remodeled in the atypical translocation of chromosome 922. All my acknowledgement to the Dr. Gida Landure, to the Dr. Omar Samaseku, and all the staff, and H3Africa for giving me the opportunity to present this work. Thanks.
Okay. Thank you so much, Mudibo K. Goita, for an amazing presentation. And I see that Mali is well represented here, as well as the PI, Guida Landore. And the presentation was on complex genomic rearrangement and individual telomere length in chronic myeloid leukemia. And yes, as I said, Goita Mudibo is going to be available at the end to answer your questions. Please do go ahead and tap them in the live Q&A, or rather in the discussion forum as well, if you sort of find that easier to use. I'm going to move on quickly to the next presentation, the third one, by Hannah Nyako, who's going to be presenting on genetics of breast cancer subtypes and race/ethnicities. Without any further ado, I'm going to hand over to Hannah to give a presentation. Hannah, go ahead.
Good day, everyone. I'm Hannah Nyako from the Kwame Nkrumah University of Science and Technology. And today, I present current findings of my MPhil work on the genetics of breast cancer in diverse ethnicities. By far, breast cancer is reported as the most commonly diagnosed form of cancer worldwide. In 2020 alone, 2.3 new cases of breast cancer were recorded, and 11.7% of all currently diagnosed cancers are actually breast cancers. Now, due to advances in early detection and improvement in treatment, breast cancer survival has increased substantially over the last 3 decades. It is reported to have an overall 5-year success rate of 90%. Sadly, this statistic does not reflect the true characteristic of the disease outcome within diverse populations. Particularly in Africa, Sintengo reported that the 5-year survival rate of breast cancer in Africa was approximately 53%. And these disparities have generally been attributed to low patronage of screening services, resulting in late-stage presentation of the disease. However, studies have shown that the disparity between race or ethnicity could even remain, even after adjusting for the socioeconomic statuses, treatment differences, and age at diagnosis. This therefore suggests that variations in tumor biology in diverse races or ethnicities may contribute to this disparity we see in prognosis. And it establishes the fact that there is the need for investigating population-specific genetic determinants of the disease. Therefore, in this study, we sought to determine the population-specific differences in the molecular characteristics of breast cancers across several populations. So for this study, we selected data sets, or we selected RNA set data sets with or without whole exome sequence data sets. And we considered only samples with race or ethnicity information. In total, we used 155 samples in this study, which consisted of 7 different ethnicities. This diagram here illustrates the workflow. So within each population, samples were grouped by subtypes, as presented in Figure 4. A clustering analysis was then performed to assess the biases and distribution of gene expression profile. Gene biotypes identified in our differentially expressed set included long non-coding RNA, microRNAs, protein coding genes, and pseudogenes. And for functional enrichment analysis, we only included protein coding genes. The gene set enrichment was also performed for each population by subtype. And here we present the top 10 activated and suppressed genes. Generally, upregulated genes were largely enriched in the activation of cell cycle processes and DNA replication processes, while downregulated genes were largely enriched in metabolic and homeostatic. In contrast to previously observed gene terms, cell cycle processes and translation and transcription processes were found to be downregulated or suppressed in the CATA group, whilst processes involving nervous perception and cellular signaling activities were activated. This heat map shows the expression profiles of the top 100 differentially expressed genes. The differentially expressed genes were then passed on for a weighted gene co-expression network analysis. First, to relate the traits to the weighted gene correlation network, a binarized file was created for representation of each population. Figure 13 here shows a sample dendrogram and trait heat map with traits as the various populations. These were clustered together and assigned model colors. The dissimilarity was based on previously calculated topological overlaps, where y-axis is the distance determined by the extent of topological overlap. So in general, we identified 13 gene models from our differentially expressed genes. To identify the correlation between the ethnicities and the models, we calculated the correlation and the significance with relation to the correlation. And then found out that the light cyan model was positively correlated with the Kenyan, Kata, and Chinese groups, and negatively correlated with the other groups. In the green model, on the other hand, the Kenyan and Chinese groups were found to be negatively correlated with the green model. We further identified the hub genes in both green and light cyan models by setting a KME threshold of greater than or equal to 0.7. 30 hub genes were identified in the green models, and these were found to be associated with estrogen receptor signaling processes. In the light cyan model, on the other hand, we identified 8 hub genes, which were later annotated as pseudogenes. So in this study, DEGs were largely enriched in the activation of cell cycle processes and DNA replication processes, except for the Kata group, where we identified otherwise. Hub genes inversely correlating with the Chinese, Kenyan, and Kata groups were identified to be associated with the estrogen signaling pathway. that would like to note that the correlation between the model and group here might have been influenced by the breast cancer subtypes rather than the ethnicity, since both the Kenyan and Chinese groups included only triple negative or basal-like subtypes. Thus, we conclude that no distinct classifications were identified by ethnicity. In further studies, we'll be assessing the possible population-specific variations by performing an integrative analysis of gene expressions and genomic alterations. And this analysis is currently ongoing. Thank you for listening, and these are my references for this. And I would like to acknowledge my project supervisor, Dr. Samson Pandam Salifu, the Fogati NIH for supporting this project, Waslid Bree, and the National Institute of Mathematical Sciences. Thank you all for listening.
Thank you so much, Hannah, for that amazing presentation as well. And I think there's something in your conclusion that you said about no sort of classification by ethnicity, and I think I'm gonna wanna ask you about that, particularly when it comes to breast cancer, because I thought there was sort of like some ethnic, racial disparity. But I mean, we'll talk about that during Q&A. And just to again encourage our attendees that you can ask your questions in the live Q&A or in the discussion forum as well. Give your comments. What exactly did you enjoy about the presentation? What specific questions do you have for who? So do say at Modibo, I have a question. At Hannah, I have this question or comment for you. Without wasting any more time, I'm going to move on to the last presentation. It's not the least, but it's the last one according to the order of today's work, because we have about 4 presentations. And the name of the presenter is Clarice Musana Baganwa. And the title of the presentation is Trauma-Related and Neutral Force Memories and Associated Biological Markers in Patient with Post-Traumatic Stress Disorders Following Exposure to the 1994 Genocide Against the Tutsi. Without further ado, I'm going to hand over the mic to Clarice to give the presentation. Thank you.
Good afternoon, everyone. Thanks for the opportunity to present in 18th H3Africa conference. I'm Clarice Msana-Baganwa. I'm an epidemiologist and biotechnologist by training, and now pursuing my PhD in epigenetics and bioinformatics. In today's talk, I'll be discussing with you about trauma-related and neutral force memories and associated biological markers and intergenerational effects in PTSD patients following exposure to 1994 genocide against the Tutsi in Rwanda. This project is being implemented at the University of Rwanda in collaboration with Radboud University, University of South Florida, and Bridge University of Brussels. Forced memory is defined as a phenomenon where people recall things that were not presented to them or record it differently from the way it happened. This Rodiger-Delmont paradigm is one of the paradigms which is used in measuring the forced memories, and this paradigm sees these forced memories as source monitoring errors, which are produced when there is a presentation of series of words which are semantically related to the real world, and then they conjure up in mind of a participant. According to the scientists, first memories are part of the working memory and can be triggered from anxiety and stress. PTSD is considered as a disorder that also affects memory because people with that condition, they are troubled by recurrent intrusive distressive memories of traumatic events. Science has shown that there's an impact of associative activation mechanisms in the linkage between psychopathology and forced memories. And mostly the forced memories are produced when there is associative material which has been presented. So according to the researchers demonstrated that there is a link between war-induced PTSD and dissociation, And also these dissociative symptoms are positively correlated with susceptibility to false memories on DRM task. PTSD group firstly recognized more critical role than other trauma-exposed participants with DRM task, according to the different scientists, but these discussions are under debate. Other scientists report that PTSD patients recall fewer words than participants without PTSD. Although several studies have been exploring the linkage between trauma-induced PTSD, there are many discrepancies between the findings on correct recalls of word and trauma-related cognitive tasks from DRM task. And there is no study which has been implemented in the context of Rwandan genocide with transgenerational effects on maternal stress. On the other side, at biological level, there are different biomarkers which have been reported to contribute in cellular stress response, such as HSP70, HSP90, and HSP27, cortisol, and others like glucocorticoid receptors. These biological markers have been reported really to be important in cell survivors, and so we hope to study these biomarkers in our ongoing studies in the Rwandan genocide survivors population to understand the level of expression of these proteins in non-traumatized population and also traumatized population. So our hypothesis is that PTSD patients may have a particular impairment in source monitoring and towards tendency of higher production of false memories on DRM lists in the context of genocide exposure. There are other different hypotheses being tested in this project, like, as mentioned, level of expression of mineralocorticoids and the mentioned proteins, as well as glucocorticoid receptors. We assume that they differ between non-traumatized population versus traumatized exposed population and their offspring. We are also testing if there's a correlation between level of corticosteroid receptors and examined heat shock proteins, and this correlation may differ between the exposed group versus non-exposed group. We also want to test if there's association between DNA methylation and memory impairment, as well as intergenerational effects of PTSD and trauma among Rwandan genocide survivors. So our motivation here is that according to the global statistics, 70% of mental disorders contribute to global disability adjusted life years, which brings to the high level of burden of PTSD, like it is ranging at 3 to 4%, and depression at 4%. But when you look on the right side of this presentation, for Rwanda, the burden of PTSD is high among genocide survivors. It's at 28%, and depression is at 10%. So this really is motivating us to study more, to have more research questions in this population, so that we can build interventions to alleviate the burden in Rwandan population. So with this DRM paradigm, we want to use it in Rwandan context, and this may maybe decrease the discrepancies which are in available data on susceptibility to recall, critical rules, and trauma-related cognitive tasks. So the objectives is to examine the free recall and false memory rate among Rwandan genocide survivors, and to determine the association between trauma exposure and memory impairment. So as I mentioned, this is a piece of work which is linked with the ongoing project under NIH grant support and H3Africa, which is looking at epigenetics and intergenerational effects in maternal stress, where we are determining DNA methylation status following exposure to 1994 genocide against the Tutsi, and also try to illustrate the intergenerational effects and epigenomic changes associated with PTSD and genocide. Methodology of this part, we are using DRM test, as I mentioned, and the DRM test is applied with the presentation of list of words that are associated with trauma with also non-studied critical roles. The stimuli is 20 list of Rwandan words. Each list contains 10 words associated with one non-presented word. The 20 lists are divided in two sets, trauma-related words and neutrals. So the study design, it contains case and control groups. We will be having 150 PTSD cases, 150 resilient, and also 150 control group. Those who were not exposed to genocide, who were outside of Rwanda, women and their children during 1994. As I mentioned, the study tool is this Rodiga MC Dermot effect, and we generated our own trauma-related words and neutral words based on DRM lists which are available. So in our pilot phase, we did it with on 36 participants, and we wanted to test the internal validity of the tools to be used. And these participants were students and staff members at the University of Rwanda. And then we added women and men at Kibagabaga Hospital who were chosen randomly. And this pilot has taken between 27 to 58 minutes. We did also pilot phase 2 to make sure that to test free recall and free recognition on generated lists. And this pilot was implemented at the same site, Kibagabaga Hospital in Bugesera, and a total of 50 participants were tested in 3 days. All participants were at Aheza Healing Center, and the time to complete the task was between 40 to 60 minutes per participant. So some of the findings that we've seen, we've seen that the list of words which were presented to the participants behaved to not be remembered easily and were not really associated between them. So here we are thinking that maybe culture and language are suspected to play a major role in DRM tasks within Rwandan context, but this needs to be confirmed at the larger population. The same with pilot 2, where we wanted to test the free recall and forced memory rate. We found that the free recall and forced memory rate was very low for all 20 lists which were tested. Only 2 words were forcibly recalled. And so it shows that the free recall was very low, and also the recognition was not well taken. On the average of these lists recalled, 4 words were remembered per list. So these are the findings of the pilot, and we are revising now the tools according to the findings from the pilot to really increase the validity of the tools that is going to be used on the larger population. So the next step is that we want to revise the tool by taking into consideration of those culture and linguistic factors. And also we anticipate to organize community engagement on memory quality study. And then we do data collection for the entire population for the 450 sample sets that we are anticipating to have by September to December 2021. So I acknowledge support for my supervisors, Professor Leon Mutesa, and Dr. Stefan from University of Rwanda, Professor Monica and Professor Derek from University of South Florida, Professor Beno and Professor Erno from Radboud University, and Professor Rose Gemini from Vrije University, Brussels. Of course, we appreciate support from NIH and H3Africa to be able to implement this kind of project. I thank you very much for your attention.
Okay, thank you so much for that, Clarice. I think you just gave me some memories about when I visited Rwanda in 2018 during a consortium meeting. When I visited Rwanda in 2018 for a consortium meeting, and I got to go to the Genocide Memorial with some of my colleagues and the locals there as well. And I think that was one of the biggest discussions around epigenetics and PTSD and the trauma that sort of resulted from the genocide, and I think it's quite important work as well. So thank you so much for this presentation. Very informative. We have a few questions. I think we do obviously not have a lot of time, and I just want to also apologize. I just want to also apologize on behalf of presenters who cannot join due to technological challenges. I mean, as we know, because of COVID, this is obviously now sort of the new normal where we do a lot of our meetings and engagements virtually. And obviously there are pros and cons to that. And I think the pros are we get to be safe from COVID. We get to make sure that we save money as well because we're not spending as much to travel. But I think the cons are technology does fail us, or I mean, Wi-Fi sometimes doesn't work. There's load shedding in some part of the world where I am, particularly we suffer from electrical outages and stuff like that. But yeah, I think I see my two presenters now and I'm gonna give them the questions. So I just want to apologize on behalf of Clarice as well as Linda. So if you have any specific questions for them, please kindly send an email to them or type in the chat live Q&A. They'll be able to then obviously engage you at a later stage, but obviously they have to type instead of speak because they're having technological challenges. But we do have amazing presentations or presenters from Hannah and Goita, who I've met actually before, both of them during a consortium meetings. I will direct a few questions to you that I see in the chat. There's a few for both Modibo and Hannah. I'll start with Modibo. Modibo, there's a question from Anonymous that's saying, very interesting. I know telomere length decreases with age. The question is there any information about how this translocation and change in telomere length interacts with the aging process? Goita? Modibo, I can't hear you. Is it just me who can hear Modibo? Okay. Because of the time issue, I think I'll just adapt quickly to go to Hannah, and we'll go back to Modibo when he gets back, because as I said, technology can be challenging sometimes. So please do bear with us, and thank you so much for attending. Hannah, I'll ask you a question from Anonymous. It's saying, did you examine any gene and environment exposures, including psychosocial exposures? Also, were these racial and ethnic groups self-identified? Thanks for your presentation. Hannah, go ahead and give your answer. Hannah? Hannah, I can't hear you. Hannah, we're also having a challenge with you.
Yes, hello.
Yes, we can hear you, Hannah. Go ahead and give your answer. Thank you.
Okay, so I can't hear you now, but basically for this particular study, we only selected samples where the information concerning their ethnicity or race was actually attached. So that was the samples we used or the data sets we selected. And with regards to examine the gene and environmental exposures, we didn't examine or we didn't look out for those factors. That's if environmental factors actually played a role in there.
Yeah, thank you so much for that answer. Hannah, can you hear me? Hannah? Because I have another question for you. Can you hear me?
Yes, I can hear you now.
Okay, perfect. There's another question for you. What is the disadvantage of the functional method you used? There's two questions actually. So the second one is, was it a ranked gene set method? That's from Anonymous as well.
Okay, so yeah, I used around gene sets for the enrichment analysis. For the enrichment analysis, I basically used the cluster profiler to do a gene set enrichment analysis. But the disadvantage. Okay, so far I had not identified any disadvantage in there, but yes, I went with a rank gene set.
For those answers, Hannah, I'm just now gonna try Mudibo again. Mudibo, can you hear me?
Yeah, I hear you.
Okay, I can also hear you. So I'm glad that you were sorted out in the background. So there's a question from Michelle Skelton, and it goes, Mudibo, can your variant discoveries lead to clinical treatment, and how?
Okay, currently, as I say in the presentation, the patient is on the imatinib, that's a tyrosine kinase inhibitor. And with our research, we didn't see any kind of this translocation, but some translocation only with 922 and 16 has been effective on nilotinib, and we need now to move a patient on the nilotinib to see if he respond about yeah.
You disconnected due to technological challenges. It's from Anonymous saying, Mudibo, very interesting. I know telomere length decreases with age. Is there any information about how this translocation and change in telomere length interacts with the aging process? That's from Anonymous. Mudibo?
Okay. Recently, actually, I can say that if this translocation is related to age, where long age or to short age, actually I can say that. But we aim to investigate more on this case and give data about it before publication on the journal.
Sorry, I'm just also, while I'm listening to you, I'm reading questions that are in the discussion forum and Q&A as well. I'm seeing there's a question from Ali. Ali, you directly had a question for Clarice, and due to some challenges, Clarice cannot sort of sign on live. So please do, and I see there's a question also from Segun for Clarice. So for those of you who want to speak to Clarice, please do find her somewhere in the chat. And I'm sure she'll be able to answer you, or rather email her directly. Apologies for that. Just general questions for the both of you then. I think obviously we had specific questions for the two of you, but I think now it's just generally about your work in doing this research and working on the topics that you're working on. What have you both learned? I'll start with Hannah. Hannah, what have you learned in this process?
Okay, so basically, I think you must have mentioned something about the having differences between the populations, or clear genetic differences between populations. So actually what I learned was my findings actually concurred with one other study where they found they identified no differences based on the expressions between the population. So that was my finding here. But then in the next study, I believe there's actually some genetic form of genetic differences in there. So in the next study, or in an ongoing study, I'm considering doing variant analysis, where I'll combine that with my gene expression sets, and I believe, yes, there are differences in there. So that's what I found out lately, that we do not only depend on just one set of the omic level data, but it's actually a combination of the omic sets to actually make a great finding.
Very interesting, because I mean, I think you've taught me something as well, because I had a few assumptions about breast cancer being more sort of prevalent in Western sort of countries, but I think I've learned something from your presentation, so thank you for that. Modibo, what have you learned in the process of doing your research?
Okay, I learned from that CML is how can I say, more study was focused only on the structural abnormality in the CML, but less study was done on the single chromosome abnormality. And that we are showing with the study and learning with the study that CML is so far to be classified as as known disease that as a model of a cancer, and there are much things to do again with CML. Yeah.
I'm saying I'm agreeing with you, and I realize that my mic was muted. Thank you so much for that answer, Modibo. So just last question to the both of you, because I'm looking at the time, and we've gone over time already, but I think obviously your presentation is so interesting and educational that I think I want to keep you here for longer. So I'll just ask one more question. So, like, what do you think is the most important thing that about your work for the public to understand? So I'll start with Modibo this time around. So if you were to sort of, because I think we do research as scientists where we use jargon and bombastic words that can be confusing for lay people. So what do you think is if we were to choose one thing that as a public person who's not a scientist or doesn't work in the field of genetics, what should I understand from your work? What would be the one thing that you'd say, Modibo?
One thing to say to the public, to the general public, is that do not forgive cancer. Cancer is taking place, and it's a complex disease, and anybody can and must take care of itself about cancer. And is that I can say to the public, to the general public. Yeah.
Awareness about the reality of the disease. Thank you so much for that, Modibo. Hannah, you are the last one to answer this question. Not last, but not least as well, I guess. What do you think from your research and from your work do lay people or the public have to understand?
Okay, I would say that we are all actually quite different in the way we, let me talk of in the aspect of genomics. We have very different genomic makeups or genetic makeups from one group of people up onto another. So that means it's very essential to actually make sure there are representations of all these groups when we are having such analysis. And yes, that actually comes down to the era of precision medicines, which this kind of studies are actually, makes them necessary.
Very graceful and comprehensive answer there, Hannah. Thank you so much for the you. Thank you, Hannah, and thank you, Modibo, for great presentations and for also giving us time to answer questions right now. I do really appreciate your time and effort, and I think you did a great job. And to Clarice and Linda, apologies that technology didn't come through or didn't allow you to sort of engage live, but I see that Clarice actually is answering questions in the discussion forum as well. So Linda, if you can do the same, I'd appreciate that as well, and I'm sure our attendees would appreciate that as well. To all of you, thank you so much for attending, and see you in other presentations that are taking place at the same time. Thank you so much for your engagement, and thank you so much for making sure that African science is in good hands, because you are fellows and you are the future of African science. Thank you so much, everybody. Goodbye.
Thanks.